ఇంతవరకు పోయిందే లేదు మాకు తీసుకుని మీ సెల్లకి స్నాప్ పెడతాను జో జో
Good evening, all. We'll wait for 5 minutes and wait for everybody. Yeah, we'll wait till everybody joins and then we'll initiate the session. So, good evening, everybody. I hope I'm audible to all.
Okay, ma'am. Good evening.
Okay, so let's initiate today's session of Human Physiology, which is taken by Professor Nishikant Subedar from IISER Pune. And I'm a TA of this course, and I myself, Tanishka. Okay. So in last few classes, we have discussed about the skeletal muscles, about the cardiac muscles, how the muscle contractions takes place. Okay. In the last class, we also talk about the cardiac tissues, its blood pressure. Okay. And how the blood pressure is being regulated. Okay. So, um, when we are talking about the blood flow in our body, we have to talk about one more system of our body, okay, which is called lymphatic system. Okay, so here you can see, so from our heart of our body, the arterial system carries the blood to different body organs. Okay, so arteries get branched and then they get divided into the capillaries, and again in the capillaries the gaseous exchange takes place, and the deoxygenated blood again travels from our tissues to back to the heart in the venous system. Okay, so in between this, when the wherever the capillaries are present, okay, so when the blood is there in the capillaries, okay, so when the blood leaves our heart, it leaves with 120-80 mmHg pressure, and when it reaches, the pressure drops, and when it is there in the capillaries, the plasma of the blood oozes out from the capillaries. Okay, and then it goes to tubules, which are nothing but the lymphatic capillaries. So these capillaries, these green color capillaries which you are seeing here, are the part of the lymphatic system. Okay, lymphatic circulation system. And from our blood capillaries, the plasma which oozes out in the interstitial fluids of the cell, that is taken by the lymphatic capillaries, and then again the lymphatic capillaries collect and to form a duct, which again brings or brings it or take the back plasma to the uh, venous system, and again with the venous system blood, the plasma which is oozes out again gets incorporated in the venous system and goes back to the heart. Okay, so the heart is not directly involved. The circulation is only taking place within the arteries and the capillaries. Okay, so the smallest units are lymphatic capillaries, smallest lymph vessel first to receive the lymph. Okay, so the plasma which is oozing out from this capillaries is called the lymph. Okay, so the lymphatic capillaries take the lymph, then they form a lymphatic collecting vessels, collect from the lymphatic capillaries. Then we have lymph nodes. Okay, here these kind of lymph nodes are present, scattered along the collecting vessels, and lymph trunks are there, collect lymph from the collecting vessels, and then we have the lymph duct empty into the veins of the neck. Okay. Hmm. The microcirculation and the lymphatic system, the capillary exchange, fluid exchange, interstitial fluid, and lymph flow. So at any moment, only 5% of the circulating blood is in capillaries, but this is the most important because it is the only pool from which oxygen and nutrients can be delivered and waste products removed. Okay, so the walls of the capillaries are extremely thin, constructed of single layer, highly permeable endothelial cells, and therefore water, cell nutrients, and cell excreta can all interchange quickly and easily between the tissue and the circulating blood. Okay, so this is all our lymphatic system. Okay, so the lymphatic system represents an accessory route through which fluid can flow from the interstitial spaces into the blood. So most important, the lymphatics can carry proteins and large particulate matter away from the tissue space, neither of which can be removed by absorption directly into the blood capillaries. And this return of the proteins to the blood from the interstitial space is an essential function without which we would die within 24 hours. Okay, so whatever plasma component which is oozing out from the capillaries at the time of gaseous exchange, that needs to be returned to the heart, and that particular activity is done by the lymphatic system. So it again takes that interstitial fluid that includes the proteins and everything, and in the lymphatic vessels and ducts, it again uh, brought it back to the heart. Okay, so this is a representative image which I can show you. So this, you can see, these are arteries, these are venules. Okay, so in the arteries and arterioles, you can see the smooth muscles are there. Then these arteries are being branched and making the capillaries. Okay, then here the gaseous exchange is having, and deoxygenated blood again goes to the veins. Okay, so here only in the vicinity of these capillaries, in the interstitial space of the tissues, the plasma oozes out. You can see the movement. Okay, the plasma oozes out in this space, which is being taken by these particular cells. These are nothing but the lymphatic capillaries, the ends. Okay, these are single layered endothelial cells. Okay, so what happens when there is a uh, stretch, and there is a gap between these endothelial cells gets created, and in the gap, when there is a stretch, there is again a contraction. Okay, actin myosin filament contraction happens whenever there is a stretch. So when that contraction happens, the fluid which is come between these endothelial cells draws within the tubules of those endothelial cells. Okay, so whenever the contraction happens, the fluid comes inside these capillaries or tubules. Okay, and then it forms lymph. It is called as lymph, and then it enters in these tubules of the lymphatic vessels. Okay, so okay, here you can see uh, these are the uh, endothelial cells of the capillary system. Okay, so you can see there are walls present. There are these kind of walls present which allow the movement of the limbs only in one direction. Okay, they carry it from the capillaries to the uh, venous system, so that it doesn't go back. Okay, that is why the walls are also present. Hmm. So as these capillaries becomes tubules, okay, and duct, so there is some arrangement or covering of smooth cells also happening. Okay, so as it again branches and collects all together, the smooth muscle covering increases. Here you can see partial smooth muscle cell coverage. Okay, when it is united, these are capillaries. Then as it is collecting, the branches are being collected, the complete smooth muscle cell coverage is happening. And because of these smooth muscle, what happens, the peristaltic movement takes place. Okay, so the peristaltic movement takes place, that means there is a wave of flow of the lymphatic fluid in a particular direction. Okay, and as this wave passes, it creates a negative pressure behind. Okay, so if the peristaltic movement is happening in this direction, the negative pressure is created in the area which allow the flow of the lymph only in one direction. Okay, so this is a schematic diagram of the lymphatic vascular tree. So this lymphatic capillaries are blind ended. Okay, please try to join the session in time. So the capillary lymphatic endothelial cells are only partially covered by the basement membrane. Button structure locate at the capillary helices, and helices anchor to the elastic component of the extracellular matrix by anchoring filament. Okay, this facilitate interstitial fluid and cellular entry into the lymphatic capillaries, and this interstitial fluid and cells can enter in the lymphatic capillary through both paracellular and transcellular routes. So this is ahead. Hmm. Okay, so yeah, so as it says that negative pressure is created that causes the excess fluid, which is interstitial fluid, taken away in the lymphatic system. Okay, so suppose in your lungs you have the capillaries and they are uh, oozing out the fluid that causes the alveoli of your lungs uh, to filled with the fluid. So that's why the lymphatic system is also very important to draw that excess liquid and takes it back in the form of lymph via the lymphatic system. Okay, so if there is a defect, there is a improper filtration is happening in the lymphatic system, the excess fluid is getting accumulated, and that causes swelling by increasing the fibrous tissue in the body. Okay, so an increase in the interstitial fluid volume is called edema, which is swelling. By definition, edema forms when the volume of the interstitial fluid due to filtration out of the capillaries exceeds the ability of the lymphatics to return to the circulation. Okay, so thus edema can form when there is increased filtration or when lymphatic drainage is impaired. Okay, that means if excess of the fluid is coming out of the capillaries, okay, then more liquid is being formed that is beyond the capability of the lymphatic system to take back as form of the lymph, or whatever is the issue, the lymphatic system or lymphatic drainage is impaired and it is not able to take whatever the fluid is coming out of the capillaries, and due to that there is increase in the interstitial fluid, that is nothing but swelling. Okay. Hmm. Okay, so here also you can see, so the lymphatic system is also one of the major routes for absorption of nutrients from the GIT tract. Okay, so in our GIT tract, in our intestine, these are the villi, and in this villi also we have the blood supplies. Okay, so these are the capillaries, you can see these are the villi, these are the capillaries, and inside this you can see this peach color uh, tubule like structure, which is central lacteal. Okay, it is called central lacteal. Okay, so this central lacteal, so whatever food is being absorbed by our GIT tract, so the amino acid and glucose are uh, taken with the blood to the hepatic portal system to the liver. Okay, so amino acids and glucose are taken by the blood to the liver via the hepatic portal system. Okay, hepatic portal system for storage and absorption. Okay, however, the fats are not taken by this hepatic portal system. The fats are taken by this central lacteal. Okay, so this um, absorption of virtually all the fats in the food. Indeed, after a fatty meal, the thoracic duct lymph sometimes contains as much as 1 to 2% of the fat. So the fat absorption work is also done by this lymphatic system of our body. So the lymph nodes in the structure we saw lymph capillaries and there are nodes present. So this is the structure of lymph node. Okay, so the lymph node removes the debris and pulls the fluid for dangerous microbes. Okay, so these lymph nodes are filled with the lymphocytes. Okay, so here you can see there is afferent lymphatic vessel, and then so there are labelings are there. So first one is this, okay, which is lymphatic venous anastomosis. Second is the node blood supply. Okay, this one. Then we have third one, which is the afferent lymphatic vessel. This one, the afferent lymphatic vessel. Fourth one is the efferent lymphatic vessel. Five is lymphanogen, and six is a lymph node. Okay, and these are all taking it back to the pulmonary lymphatic capillaries and putting it back to the heart. So these lymph nodes, which are filled with the lymphocytes, they have the work to remove the debris and police the fluid for the dangerous microbes. Okay, yeah. So there is this particular worm which is Vaucheria worm. So this worm sits on the lymphatic system and causes a defect. Okay, and because of that defect there is an impairment in the lymphatic drainage system which causes the swelling, that edema that we just now discussed. So this disease is called elephantiasis. You can see the legs, they have swollen a lot, and this disease is called elephantiasis. Occurs in the presence of this microscopic thread-like parasitic worm, Vaucheria bancrofti, which is transmitted by mosquitoes, and the adult worms only live in human lymphatic system. The parasite infect the lymph nodes and block the flow of the lymph throughout the body. Okay, and this result in chronic edema, most often noted in lower torso, typically in the legs and genitals. Okay, so I hope this lymphatic system part was clear. Okay, let's see the renal function. So let's talk about the kidneys. So the kidneys have several functions to perform. So the major function we know is the filtration of the blood, but along with that it has several different functions. First one, if we'll see, regulation of water, inorganic ion balance and acid-base balance in cooperation with the lungs. Removal of metabolic waste product from the blood and their excretion in the form of urine. Removal of the foreign chemicals from the blood and their excretion in the urine. Gluconeogenesis, production of hormones, erythropoietin, renin, and also this particular one which influences calcium balance. Okay, so in the last class we have also discussed about renin enzyme that controls the formation of angiotensin and influence the blood pressure and sodium balance. Okay, so the kidneys are primary means for eliminating waste product and metabolism that are no longer needed by the body. So these products include urea, okay, from the metabolism of amino acid, creatinine from the muscle creatine, uric acid from the nucleic acid, and end products of hemoglobin breakdown such as bilirubin and metabolites of various hormones. So these are the majority of the components that needs to be excreted out by our body, and that is being filtered out by the kidney. So these waste products must be eliminated from the body as rapidly as they are produced. So the kidney also eliminates most toxins and other foreign substances that are either produced by the body or ingested, such as pesticide, drugs, or food additives. So urea from the metabolism of amino acid, creatine. So these are the list. Urea we are getting from amino acid, creatinine that is formed by the muscle creatine, uric acid from the nucleic acid breakdown, bilirubin and product of hemoglobin breakdown, and metabolites of various hormone and drugs. These are the things that are being removed by the kidney by the filtration of the blood. Okay, so this is the renal system. Okay, so here you can see the two kidneys of our body. Okay, so the where the kidney takes the blood, that is artery, and that artery is referred as renal artery. Okay, so this is a renal artery which is carrying blood to the kidney from the aorta, and this is a renal vein which is carrying the impure blood from the or the filtered out blood from the kidney back to the heart. Okay. Oh, discuss later. Yeah, so this is the structure of a kidney. So we have the bean shaped kidney, and if you take a section of the kidney, you'll see something like this structure you'll see. Okay, so this is covered with a thin layering of connective tissue which is called as renal capsule. Renal capsule. Okay, and then inside this you will see this portion is a renal cortex portion. Okay, the outside, this entire portion, the outside portion, when you see from the outside, the outer portion is the renal cortex. Okay, and in between these structure which is a little dark, they are referred as renal medulla. Okay, these are pyramid shaped structure, these are renal medulla, and these are also renal pyramids, these are referred as renal pyramids. Okay, so the base of the pyramid is facing the cortex, and the apex of the pyramid, which is uh, inward, that is facing this space which is called renal pelvis. Okay, so this space is called renal pelvis. Okay, and then we have, okay, so this is renal pelvis, and um, inside the function, inside this kidney we have the functional unit of the kidney which performs the filtration process. Okay, and after the filtration, all the urine gets collected, okay, by these structures. It comes in this renal pelvis, and from renal pelvis it goes to the this particular duct which is called ureter, and beyond this it goes, both the ureters open into the urinary bladder. Okay, where the urine is stored. Yeah, so yeah, these are the cortex portion which is inverted in the medulla, that is called the column, this renal columns. Okay, and here only we'll find, in this particular portion we'll find the functional unit. Okay, the renal cortex, renal medulla. Okay, they form a particular functional unit of the kidney which is nothing but the nephron. Okay, you can see one nephron is being shown here. Okay, so these are the nephron which does the function of the filtration. Okay, so if we'll see next one. Yeah, so here you can see, so as we have seen, uh, the renal artery, okay, from the aorta the blood is coming, and it is the artery which is taking the carrying the blood to the kidney is called renal artery. Renal artery further gets divided and makes these segmental arteries. Okay, so here you can see the division of the renal artery, and it is making segmental artery. Okay, so segmental artery further gets divided and forms the interlobar arteries. Okay, these interlobar arteries you can see. Okay, and then these interlobar arteries further divided and make the arcuate arteries. Okay, and these arcuate arteries only enter the our nephrons, the unit of uh, the unit of the kidney, the functional unit of the kidney. So these arcuate artery, so the particular uh, way the tube of the arcuate arteries that enters the nephron is nothing but this particular afferent arteriole. So here you can see the arcuate artery is present, which is divided, and this portion of the arcuate arteriole, the fragmentation which enters the uh, nephron, is the afferent arteriole. Okay, so a nephron contains of the afferent arteriole, which is further branched to the capillary network. Okay, and these capillaries, and then again here the filtration happens. Okay, so these is nothing but these networks, okay, of the capillaries is called glomerulus. Okay, and this glomerulus is present in this particular capsule, is called the Bowman's capsule. Okay, this one you can see yellow thing is the Bowman's capsule. So in the Bowman's capsule we have the glomerulus present, and in the glomerulus only the filtration of the blood takes place, which is coming from the afferent arteriole, and after the filtration the remaining blood goes to the afferent arteriole. Okay, then the afferent arteriole again divides and makes this peritubular capillaries. Okay, this afferent arteriole. Okay, so this afferent arteriole is coming out, and these from here the filtration is happening, and from the Bowman's capsule it is going to the proximal convoluted tubule. Then we have this loop of Henle, and then we have distal convoluted tubule. Okay, where after the filtration the reabsorption and filtration process will take place, and then again all together it goes to a collecting duct. Okay, this is the collecting duct, and from collecting duct all those collecting duct opens and pour this filtrate to this ureter. Okay, so this is the entire arrangement. Okay, I hope it's clear to everyone. So yeah, these are the, here it is a little more spread and clear. So you can see here the afferent arteriole and efferent arteriole of the glomerulus, and this is the Bowman's capsule, which from the Bowman's capsule this is a proximal convoluted tubule, which is again making a long tube of the, which is called loop of Henle. Then it is again making a distal convoluted tubule, and this distal convoluted tubule is opening into common collecting duct. So here also one more nephron is there. Similarly, afferent efferent arteriole, glomerulus, Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and again opening in the collective duct. So many such distal convoluted tubules of different neurons opens in this particular collecting duct. Okay, so such we have many different kind many in number such neurons are present in a kidney. Near a million neuron of such are present in the kidney of human's body. Okay, so we have seen this. So here you can see there are two types of neuron you can see. Okay, sorry, nephrons you can see. So this portion of the kidney, this portion is the cortex portion, the outer portion of the kidney, and here this one is the medulla. Okay, in the medulla also they have divided outer medulla and inner medulla. Okay, so there are two types of neuron. One neuron where the maximum portion is present in the renal cortex only, and a part of the loop of Henle is entering into the outer medulla. Okay, so a little portion is entering into this medulla. However, in this kind of neuron you can nephron you can see this uh, majority of the loop of Henle is coming into the renal medulla only. Okay, so these are the two types of nephrons which are present in the kidney. Majority of the nephrons are of this type. Okay, and this type of neuron where the loop of Henle is present in the medulla, Okay, in the renal medulla of the kidney, the nephrons are called juxtamedullary nephrons. Okay, these are less in number, these are more in number. So these are the two different kinds of nephrons which are present in the kidney. So this we have already discussed. Okay, so the renal circulation is unique in that it has two capillary beds. Okay, the glomerular and the peritubular capillaries. Okay, these are the peritubular capillaries and these are the uh, uh, glomerulus capillaries, which are arranged in a series and separated by efferent arteriole. One more thing which is interesting about these glomerular capillaries, these are the only capillaries which are uh, like getting divided. An artery, usually what happens in artery divides into thin capillaries. Okay, thin capillaries, and then here the gaseous exchange will takes place, and then again these capillaries uh, will mix in a venule. Okay, so usually the capillaries, at one end it has an artery, another hand it has a veins, but these are the special kind of arteries, capillaries, which both ends are a type of arteriole only. Okay, so the venous system comes much later when the peritubular capillaries arise. Okay. So these help to regulate the hydrostatic pressure in both sets of capillaries. So high hydrostatic pressure in the glomerular capillaries is about 60 mmHg. Okay. So when the blood enters in the renal artery, it enters with a pressure of 60 mmHg. Okay. So with 60 mmHg pressure, it enters and causes a rapid fluid filtration. With that pressure, when the blood enters in the glomerular capillaries, it causes a rapid fluid filtration rate. Okay, and much lower hydrostatic pressure is there in the peritubular capillaries. As it travels from here to here, the peritubular capillary, the pressure drops to the 13 mmHg, permits rapid fluid reabsorption. So the pressure, the dynamic pressure, is a very interesting part in the nephrons. Okay, so here the pressure is very high, which is helping them, the capillaries, to do the filtration of the blood. Okay, and as it leaves this Bowman's capsule, these capillaries, the pressure drops to 13 mmHg, which helps in the reabsorption of the lost material. Okay, so suppose the filtration has caused um, losing a very important substance of the blood, then it gets reabsorbed with the pressure which is visible for the reabsorption in the peritubular capillaries. Okay. So the capillaries of the glomerulus are fenestrated, which allows large amount of solute rich fluid to pass. Okay. Remember, there should not be high amount of protein in the urine. So the inner layer of the glomerular capsule contains highly modified branching epithelial cells called podocytes. Okay, so the glomerular uh, capillaries are capillaries which are receiving the blood with a very high pressure, 60 mmHg, and because they have to do the filtration and to handle such a high pressure, because if you remember in the other parts of our body, when the capillaries faces very low pressure, sub 3 to 4 mmHg only, but these capillaries are facing very high blood pressure, which is 60 mmHg. So that is why these cells have an extra layering of the cells, which is called podocytes. Okay, which is podocytes, which we'll discuss further also. So this podocyte terminates in foot processes, which surrounds the basement membrane of the glomerulus. So the cleft between the foot processes are called filtration slits, and this is where the filtrate enters the capsular space. So the glomerulus also contains smooth muscle-like cell called glomerular mesangial cell. These cells help regulate the blood flow in the glomerulus by contraction and engulf macromolecules that get hung up during filtration. Okay, so here you can see these glomerular capillaries. This is afferent arteriole, this is efferent arteriole. Okay, here the glomerular capillaries are there, and here you can see the covering of these capillaries are of podocytes. Okay, these glomerular capillaries covered by podocytes. These are the epithelial cells that make up the visceral layer of the glomerular capsule. Okay, and this is a parietal layer of glomerular capsule, proximal convoluted tubule, and this is the space. Okay, this is a much detailed structure of this capillary. Okay, this is the fenestration, the pores you can see here. Okay, these are the uh, cytoplasmic extension of the podocytes, and this particular one is the filtration slits, the spaces between the food processes covered by very thin slit diaphragm. Okay, where the filtration takes place. Okay, if you'll see a dissected section, this is the inner layer, the endothelial layer of the capillary, which is, this is the nucleus of that endothelial cell. Okay, this there the RBC is passing, and this is the filtration membrane. Okay, this is the filtration membrane which is permeable for the filtration. Okay, and these are the podocytes. So these podocytes are something like this. These are not sitting like this, they are sitting something like this to the uh, uh, glomerular capillaries, providing them slits for the filtration. Okay, but all these different podocytes are of from same cells only, a single podocyte cell, but their arrangement is such a way that they provide a filtration slit for the filtration of the blood. Here you can see these are the podocytes, and this is the food like progression, which is nothing but the pedicels. These are called as pedicels. This is the glomerular capillaries. Here the blood is flowing. Okay, and this is again the same section. Afferent arteriole, efferent arteriole, these are the capillaries, which is covered by the podocyte. Single podocyte is covering, and again same thing. So three layers, which is fenestrated endothelium of the glomerular capillaries. Okay, and then we have the visceral membrane of the glomerular capsule of podocytes and intervening basement membrane. This is the plasma. Okay, and this is the filtrate in the capsular space. Okay, this one is the capillary endothelium, this one is endothelium, then we have the basement membrane, and these are the food processes of the podocyte of the glomerular capsules. Okay, and these are the fenestration or the pores. Okay, so basement membrane meshwork of the collagens and the proteoglyans fibers that have large spaces for filtration. They also hold strong negative electrical charge. Okay, and the layer also has a negative charge that restrict filtration of plasma proteins. Okay, and because of this negative charge, the plasma proteins are not being filtered, only the small proteins molecules are only filtered. Okay, so if you have, okay, so this is the RBC. In this part we'll have the RBC, in this part we have the filtrate. Okay, which we call glomerular filtrate. Okay, so in this part from the RBC the plasma is being filtered. Okay, so in if you remember, the RBC is of 55% of hematocrit. Okay, so if you will see, take 10 ml of or 100 ml of blood, you get approx 60 ml of the plasma out of it. Okay, so when the filtration will happen, what happens, majority of the plasma tries to come out, but because of the so high rate, the plasma, all of the plasma doesn't come out. Okay, so the blood travels for the filtration many times in our body. Okay, and the rate at which the filtration is happening, okay, that is called the glomerular filtration rate, GFR. Okay, so per minute you have some 125 ml of the plasma of the RBC is being filtered, so that is called the glomerular filtration rate. Okay, and also because of the negative charge, the large proteins molecules are unable to pass the plasma in any case, and because of their size also, the permeability of those large protein for crossing or by getting filtered is very less. Positively charged dextran is filtered more freely. Here you can see. Yeah. So you can see a lot of proteins or the plasma or the electrolytes are coming. These nutrients are coming, but not all of them are being filtered. Okay. So that is why for the proper filtration, the blood travels to the kidney and to the nephron and to the glomeruli many times. Okay. So, uh, yeah. So the filterability of the substance by glomerular capillaries based on the molecular weight. So here you can see the water molecular weight is 18, filterability is 1, sodium 23, filterability is 1, glucose, insulin, the filterability is 1. Here, however, the myoglobin albumin, the large protein molecules, their molecular weight is very high. And as the molecular weight is increasing, the filterability rate is also decreasing. So filterability of the solute is inversely related to their size. Okay. So the glomerular capillaries membrane is thicker than most of the capillaries, but it is also much more porous and therefore the filter fluid at a high rate. Okay. So despite the high filtration rate, the glomerular filtration barrier is selective in determining which molecule will filter based on their size and electrical charge. Okay, so this is the table showing with increasing size, the filtration rate decreases. Okay, so this is so as the molecular weight of the molecule approaches that of the albumin, the filterability rapidly decreases, approaching to zero. So glomerular filtrate, the first step of the urine formation. So the composition of the glomerular filtrate. Urine formation begins with filtration of large amount of fluid through the glomerular capillaries into the Bowman's capsule. However, the proteins are not filtered. Most salts, organic molecules are freely filtered. Exceptions are few low molecular weight substances like calcium and fatty acid because they are partially bound to the plasma membrane. Yeah, so this is the GFR I was talking about. So glomerular filtration rate is about 20% of the renal plasma flow. Okay, so yeah, so renal plasma is what? If I'm saying 1000 ml of blood is being flowed, the renal plasma amount will be 550 ml, or you can say approx 600 ml of that will be plasma. Okay, there is a term, I think last session also I mentioned, hematocrit. Okay, so when you take the blood, okay, in a tube and spin, suppose you put it under centrifugation, you spin the blood, then if you take 10 ml of blood, out of 10 ml you will see the heavy RBCs will be settled down and the plasma will be able to see a layer of plasma upside the RBC. Okay, so you have plasma and you'll have RBCs. Okay, so uh, if this is 100, out of that 45% are RBC, 55% is plasma. So this is the hematocrit. The RBC, the percentage of RBC in that total blood is 45%, that is hematocrit. So it is 45% hematocrit our blood should be. So 55% of that is plasma, and that so it is flowing in a renal artery. So in the renal artery, out whatever plasma is being flowed, that is called the renal plasma. Okay, so if 1000 liter of blood, 1000 ml of blood is being flowed to the renal artery, 600 ml would be the renal plasma. Okay, and this glomerular filtration rate is 20% of the renal plasma flow. Okay, so the glomerular capillaries have a much higher rate of filtration than most of the other capillaries because of the high hydrostatic pressure. Okay, combined blood flow through both the kidney is about 1100 ml per minute. Okay, from both the kidneys, the blood flow is 1100 ml per minute, and about 20% of the plasma is filtered through the glomerular capillaries. Okay, so in human, the GFR is 120 ml per minute. Okay, so if you say that 1100 ml of blood will be the plasma, sorry, 1100 of total blood. So out of 1100, the plasma or the renal plasma would be only will be 600 ml. Okay, and from 600 ml, the renal flow rate is 20%, right? The filtration rate, sorry, the renal filtration rate is 20%. So if your glomerular uh, capillaries are having 600 ml of the renal plasma, only 20% of that plasma is able to get filtered. So out of 600 ml, 20% of the 600 ml is 120 ml. So in a minute, if 1100 ml blood is going to the glomerular capillaries, it is having 600 ml of plasma, and 20% of that plasma, which is 120 ml, is filtered per minute by both of the kidneys. So in humans, the glomerular filtration rate is 120 ml per minute. Okay, so then you have this. So after the filtration takes place, then you again have the reabsorption chance. Okay, this one. So first is the glomerular filtration. This is glomerular filtrate in the Bowman capsule which coming. Second is the reabsorption of substance from the renal tubule into the blood. Okay, again reabsorption will happen. Whatever excess thing is being filtered, then again get reabsorbed into the blood. Third is the secretion of the substance from the blood into the renal. Some part again gets secreted because of the proximity, and altogether the urinary excretion rate is filtration rate minus the absorption rate plus secretion rate. Okay. Okay. So these are the four scenarios which is being shown. So there are certain what to say, substances which which gets filtered. So this is your afferent arteriole. Here glomerular capillaries are there, then they make the filtrate, and whatever the filtrate is going, it is all, whatever filtration is happening, all of that chemical is going to the urine. Nothing is being reabsorbed. Here you can see no reabsorption is happening. And what, okay, then you have okay, so this is substance A. Substance A is waste products like creatinine. Okay, so if your blood is carrying the creatinine, and whatever creatinine is being filtered, it never reabsorbs. Okay, and it entire thing which is being filtered, it goes to the urine. Okay, then we have some components such as electrolytes. Okay, so electrolytes, what happens, filtration takes place and also partial reabsorption takes place. So here you can see for the efferent arteriole, here the in the glomerulus the filtration takes place, and whatever when it goes to the loop of Henle, here some part is also being reabsorbed and goes carried back to the blood. Okay, so some electrolytes, for example sodium, uh, and chloride ions, these electrolytes are highly or highly not partially gets absorbed. um, okay, here it is written, potassium and this hydrogen ions excreted in the urine, urea, creatine, uric acid, and urates poorly absorbed and excreted in large amount. Foreign substance and drugs poorly reabsorbed and actively secreted in tubules. Electrolytes like sodium and chloride and bicarbonates are highly absorbed, and nutritional substances like amino acid and glucose are completely reabsorbed. Okay, so here see amino acid and glucose. Okay, glucose never goes in the urine. Okay, whatever glucose is being filtered, all of it gets reabsorbed, glucose and amino acid. So the third scenario, which is a type of substance C, which is includes amino acid and glucose, whatever is being filtered here, everything is getting reabsorbed and uh, brought it back to the blood. Okay, so everything is being reabsorbed, and there are some substance like drug metabolites, okay, which are dangerous for the body, are actively secreted. Okay, so whatever came, it filtered, and whatever is filtered is not being reabsorbed, going to the urine, and also it nothing is reabsorbed. However, here due to the proximity of the peritubular capillaries, it gets again secreted to the loop of Henle and again goes to the urine. Okay, so these are the different kind of processes takes place according to the um, nature of the substrate that is being filtered or passed through the kidney. Okay, so here you can see glucose. Okay, so 180 gram per day amount of the glucose is filtered, and entire thing is being reabsorbed. So amount of the glucose that is excreted is zero. Nothing is being, no glucose is being excreted after the filtration. Okay, whatever is filtered is reabsorbed again and brought it back to the blood. We have bicarbonate ions. Okay, here the amount filtered 4320, amount reabsorbed 4318. So 2 milliequivalent of bicarbonate is you can say excreted. Okay, majority of it reabsorbed. Sodium and chloride, you can say if you say 150 and 180 milliequivalent of per day is excreted, majority of the component of sodium and chloride is reabsorbed. For potassium, you can see 756 milliequivalent per day is filtered, and out of that 664 is reabsorbed. 92 milliequivalent per day is excreted. Urea 46.8 is filtered, 23.4 is reabsorbed, 23.4 is excreted. Half of it is excreted. Creatinine 1.8 gram per day creatinine is filtered, nothing is reabsorbed, and everything is excreted in the urine. Okay. Okay, so this is creatinine. So creatinine is nothing but, you can say, a reservoir of high energy phosphates. Okay, so creatinine is present, and whenever there is a activity in the muscle, the creatine is rapidly converted to the phosphocreatine. Okay, and this rapid conversion of creatinine and phosphocreatinine helps in the formation of the ATP. Okay, for every muscle contraction you need the supply of ATP. Okay, many ATP supply is needed, so that's why this particular molecule is present, which is creatine. Okay, when it is in muscle it is called creatine. Okay, creatine gets phosphorylated and it is called as phosphocreatine. So phosphocreatine, also known as creatine phosphate, serve as a rapidly mobilizable reserve of high energy phosphates in skeletal muscle and in brain. And phosphocreatine can anaerobically uh, donate phosphate group to ADP during the first 2 to 7 seconds following the intense muscular or neuronal effort. Okay, and the creatinine is chiefly filtered out of the body. Here you can see creatine phosphate. It is again a spontaneous cyclization. It makes creatinine, and this creatinine, it goes to the blood, and this creatinine is filtered out of by the body. So creatinine is chiefly filtered out of the blood by the kidneys, glomerular filtration and proximal tubular secretion. Okay, there is a little or no tubular reabsorption of the creatinine. If the filtering of the kidney is deficient, creatinine blood level rise. Okay, so usually in the blood, whatever creatinine is there, uh, it should be filtered by, actively filtered by the kidneys. Okay, so the creatinine levels in blood and urine may be used to calculate the creatinine clearance. Okay, CRCL, which reflects the glomerular filtration rate. So in healthy human serum, creatinine is 0.7 to 1.2 mg per day DL. Okay, and if there is a problem in the filtration or in the kidney functionality, the blood creatinine level goes high. Okay, so that tells the level of creatinine in the blood and urine tells about the healthiness of kidney and the creatinine clearance and the glomerular filtration rate. So this is the net pressure which is applied for the filtration. Okay, as I have said, whatever renal plasma is being flowed, only 20% of that is being filtered. Okay, even though the blood is coming with a very high pressure. Okay, which is glomerular hydrostatic pressure plus 60 mmHg. Blood is coming with 60 mmHg pressure. Okay, it is because of that high pressure, it is putting a pressure outside of the capillaries to uh, uh, push the liquid outside the capillaries. Okay, this is a very high pressure which is being put. Then you have a plasma colloid osmotic pressure. Okay, plasma colloid osmotic pressure, it is a pressure applied from the outside. Okay, from the outside of the because of the proteins, the large proteins present uh, in these capillaries when blood is passing, they are creating a pressure, a negative pressure, which is called the plasma colloid osmotic pressure. Okay, and then outside this capsular fluid which is present, that is also putting a capsular hydrostatic pressure to these capillaries, that is again an inward pressure, that is why it is a negative. Okay, it's a negative pressure. So uh, this is a positive pressure and these two are the negative pressure. So all together, net pressure which is applied on the glomerular capillaries for the filtration is only 10 mmHg. Okay, so that is why I told, even though the blood is coming with a very high blood pressure, the net pressure is only 10 mmHg and only 20% of the renal plasma is being filtered at a time in a minute. This This is approximately pressure and vascular resistance. This also we have discussed how the pressure changes. Here you can see the renal artery pressure, pressure in vessel beginning 100 mmHg. Okay. Interlobular arcuate, interlobular arteries, afferent arteriole, glomerular capillaries, the pressure. Okay. So in the renal artery, the blood is coming with the average pressure of 100. Okay, it is 120/80, average 100 mmHg. When it is coming to the glomerular capillaries, the pressure is 60 mmHg. When it is leaving to the afferent arteriole, it is 59 mmHg. In the peritubular capillaries, the pressure is 18 mmHg. In the interlobular arcuate veins, it is 8, and again in the renal veins, it is 4 mmHg. Okay, so that is what the other parts of our body, the capillaries, the pressure is 4 mmHg. So that entire diversity of the pressure is faced by the kidney. So relatively high in glomerulus, strong outward force pushing fluid out of the capillaries in the glomerulus. Okay, and this glomerular filtration rate is also having a regulation. So this glomerular filtration rate is auto-regulated by the kidney. Okay, so when the blood pressure increases in some scenarios, high blood pressure, if a patient faces a high blood pressure, so what happens? To prevent the damage to the kidney, the GFR rate is not changed, even though the blood pressure is increasing after certain time, but still the glomerular filtration rate remains constant because of the auto regulation of the GFR. Okay, and that is done by the kidney. Okay, so there are two different methods, myogenic method and one more method, which is nothing but what happens, the afferent arteriole cells, which are surrounded by the smooth muscle, they are very um, important and crucial cells. So what happens, when the pressure rises, they stretch. Okay, and when they stretch, again they constrict, and because they constrict, what happens, uh, the rate of the blood flow becomes slow. Okay, so even though the blood is coming with a high pressure, because of the constriction of the afferent arterioles, the blood pressure again goes down. And that is why, again, even though the our body's blood pressure is very high, the renal plasma that flows inside the glomerular capillaries, there the blood pressure is maintained at the normal level, and that is why the glomerular filtration rate is also the constant, even in the high or low blood pressure. If the blood pressure is low, the opposite happens. Okay, the dilation happens and the pressure increase, maintaining the constant glomerular filtration rate. Okay, so other than that, one more function of the kidney is there, which is production of a particular protein or hormone, you can say, which is erythropoietin. Okay, so what happens, in some scenarios our body faces low oxygen level. Okay, there are many factors that decrease the oxygenation. It could be low blood volume, overall blood volume is decreased, anemia is there, which is very common condition in female, and low hemoglobin is there. Okay, poor blood flow is there, pulmonary diseases are there, and because of that the tissues are not able to get proper oxygen via the blood. So that is why there is a decrease in the tissue oxygenation. So to overcome this problem, what happens, if you have um, less oxygen, what you need, you want more blood. Okay, if the tissue is not able to get oxygen, it further requires more and more blood to get the proper oxygen from the RBC. Okay, so to get the more RBC, the kidney is helping and producing one such protein that is called erythropoietin. Okay, so in the kidney, this production of erythropoietin is happening, which actions on the hematopoietic stem cells of the bone marrow. Okay, so the bone marrow is the place where the RBC production takes place. So this erythropoietin, which is produced by the kidney, goes and acts to the hematopoietic stem cells, which is proerythroblast cells. Okay, and that it has the receptor for the proerythroblast cells, the hematopoietic for the erythropoietin, and these proerythroblast cells start producing RBC rapidly by the action of the erythropoietin, and that RBC goes to the tissue, gives the oxygen to it, gets tissue oxygenated, with further signals the kidney to decrease the production of erythropoietin, and that is how entirely the kidney helps in the RBC production for the tissue oxygenation. Okay, so the total mass of RBC is regulated within narrow limits, and any condition that causes decrease in the quantity of oxygen transport increases the rate of RBC production. Okay, so when a person becomes anemic, whatever the reason, the bone marrow immediately begins to produce large quantities of RBCs. Destruction of bone marrows causes hyperplasia of the remaining bone marrow. Similar phenomena operate in people living in high altitudes. Okay, so this is the content of today's. So if we'll see this image, what is this system? What it is showing? It's the lymphatic system. Okay, so what is this? Here we can see our systematic and pulmonary circulation. This is our pulmonary circulation, and this is our systematic circulation. Okay, so here you can see the capillaries network. It oozes out the plasma in the lymphatic capillaries, and lymphatic capillaries are being joined in the nodes, and these nodes are called lymph nodes. Okay, these are lymph nodes. Okay, these are the afferent lymphatic vessels, which is bringing the lymph to the uh, lymph nodes. And then we have third one is nothing but lymphatic capillaries. These are the lymphatic capillaries. And this one, second one, which is uh, bringing blood from oxygenated blood from heart to the body tissues, is systematic circulation. Okay, yeah, so this is the kidney structure. Okay, so here two regions in the kidneys section. This one is renal medulla, and what about this one? What is this portion? Yes, it is renal cortex. Okay, then we have this third one, this particular portion. What is this? Yes, yes, it is renal pelvis. Okay, so from all the collecting duct of the nephrons, they release the filtrate and the urine in this particular branches, and which collects in the renal pelvis, and it sends to the ureter, and then finally to the urinary bladder. Okay, so this is renal cortex. This is renal pelvis. What is this? This duct which is taking the, yeah, this is ureter. Okay, and this is, what is this structure? This is the, yeah, this is a nephron. This is a nephron. Okay, and we also studied two types of nephron. One, majority of the nephron where, sorry, juxtamedullary. Okay, so if the loop of Henle is present, majority of the loop of Henle is present in the renal medulla, it is called juxtamedullary neuron. Okay, juxta means close, medullary is the medullary portion. Hmm. Okay. Okay. Yeah. So these are the different uh, portions. Okay. So this particular arteriole, which is taking the blood to the glomerulus. This one, which yeah, yeah. So which takes the blood to the Bowman's capsule, that is afferent or efferent. Yeah, this one is afferent. Okay, and this one which is coming out of the glomeruli, that this arteriole is this one is afferent. This is afferent. Okay. And what is this? The capillary network in the Bowman's capsule is yes, glomerulus. Yes, it is glomerulus. Okay. Okay. And the glomerulus is present in this Bowman's capsule. Okay. And what is this structure? Yes, it is loop of Henle, where you will find the peritubular capillary. Yes. Yes. Okay. So this is a section of what? It is a section of, yeah, glomerulus capillaries. Yeah, capillaries. Okay. And these are made up of three layers. Okay. Three layers. So the inner layer, this one is what? No, no, no. The inner layer is of endothelial cells. Endothelium. Yeah. Yeah. The porous endothelium cells. Yeah. Okay. You can see the pores are there, right? And after that, the second layer is of, yeah, basement membrane or filtration membrane. Okay. Yeah, filtration membranes. This one is filtration membrane. Okay. Then, yeah, in between you can see the filtration slits are there. And what are these? Yeah, yeah. So these are of one single podocyte only. Okay. So a large podocyte remains like this, and these are the pedicels. Okay, attached, leaving some space. So these are called podocyte pedicels. Okay, correct. Yeah, fourth is RBC. Okay, so this is, what is this image telling us? Hmm. Yeah. So the RBC production by kidney. Okay. So here the factors that decrease the oxygenation. Okay. So there is a decrease in oxygenation. Then what is happening here? Second one. Yeah. So decrease in tissue oxygenation. Okay. Decrease in tissue oxygenation. Okay. When tissue oxygenation is decreases, then what kidneys are doing? Renin. Renin is for the blood pressure regulation. Okay. Renin regulates the blood pressure by production. Yes, yes. Kidney produces the erythropoietin, which acts on hematopoietic stem cells, particularly on the pre-erythroblasts. And what does pre-erythroblast cells do? It increases the, yeah, it increases the production of red blood cells, RBCs. Okay, so that further increases the tissue oxygenation. And when the tissue oxygenation is increased, there is a signal to decrease the erythropoietin production by the kidney. Yes. Okay. Yeah. I hope everything is clear. If you have any doubt, please ask me. Mm. Yeah. Sorry, your question is not clear, sir. Can you ask again? Yes. Yes. Hmm. It again goes back to the circulation. Yeah, through the efferent it exits. Hmm. Yeah. And again comes back to the for the filtration again. Okay. So continuously, so daily our body filters approx 180 liter of the blood. Okay. So whatever blood is coming, 20% of that of the that is of the plasma is being filtered. Okay, that is 120 ml per milliliter. Now whatever is not being filtered, it again goes back to the efferent uh, arteriole. If there is something that needs to be reabsorbed or secreted, that exchange will happen, and again it goes back to the circulation by renal vein. Okay, here you can see. One second, one second. Hmm. Yeah, what was your question? Sorry. Filtration permeability you are asking, I guess. This one. Filter. Yes, yes. So, glucose is never excreted in our urine. Okay, whatever. If it in the Bowman's capsule blood is coming, okay, and from that blood 180 gram per day glucose is filtered. Okay, this quantity, whatever is this quantity, it if it is being filtered and it has come to the glomerular filtrate, it again gets reabsorbed. Okay, and nothing is here. These are the models which I have shown now. So the third one, see, this one is for glucose. Okay, if glucose is coming and if glucose is being filtered also because of the high blood pressure, okay, because of the blood pressure, if it is filtered, again when it comes to the loop of Henle and in the peritubular capillary networks, it again reabsorbed by the blood. Okay, and nothing, uh, negligible amount of glucose is being lost. Okay, in the urine glucose never comes out. Okay, if there is excess of glucose, too much of the glucose, then only some amount of glucose will remain and it is not clearly reabsorbed. Otherwise glucose is reabsorbed, entire portion of the glucose is reabsorbed and does not go to the urine. Okay. Okay, anything else, anyone? Okay, if there is no further questions, we'll conclude today's session. Thank you everyone for joining. See you again next week.